So first of all, I want to thank the organizers, Liu Xiao, for giving me this opportunity to present my work and also a chance to introduce my lab, my new lab. So I joined the Center for Synthetic Immunology from is actually in Shenzhen, Shenzhen Advanced Institute of Shenzhen Institute of Advanced Technology from the Chinese Academy of Science. And the primary focus, one of the focus of my lab is to understand tumor immunology with single cell technology and trying to develop more effective and precise immunotherapy. So over the last decade, we have actually witnessed how single cell technology is transforming biological research, of course, including immunology. And particularly for immunologists, from the beginning, we characterize different immune cells based on morphology under the microscope. And then later on, we have the flow cytometry technology to distinguish different immune cells based on a few cell surface markers. And nowadays, we are actually able to use a very unbiased approach to profile the immune cells at a transcriptome level. And in that way, that we can really understand immune cell heterogeneity, differentiation, and function. And immune cells, they are quite mobile and also very diverse, and they can adapt really quickly to the microenvironment and to the environmental stimuli. And they can, in response to those stress or stimuli, they can start to secrete effector functions or start to express many cell surface markers. And so using this unbiased approach, we can really profile the immune cells at a very high resolution. And the single cell genomic community, they are also continuing to develop many different single cell technology for immunology research. For example, we can use single cell transcriptome combined with pulse trace assays to study time-dependent process in vivo. We can also study the cell-cell organization in a specific tissue, and particularly for immune cells, we know that the immune cell interaction is very, very important for immune function. For example, T cell talks to B cells, and T cells can also talk to B cells. So the community is also developing methods to look at cells that are interacting with each other. For T cells and B cells, we can also combine transcriptome profiling with Tcell receptor sequencing or PCR sequencing. In that way, we can use the T-cell receptor or B-cell receptor as an endogenous barcode to really trace the cellular fate of T cells or B cells. So last year we have developed a technology which allow us to really profile the transcriptome and also the T-cell receptor sequence from a single T cells. And this was already published last year. We use this technology to profile the T cells and also other immune cells from human cancer, and trying to understand what is the cellular heterogeneity of immune cells and what are their function and how T cells differentiate in human cancer. So we know that the immunotherapy depends on functional and also tumor-specific T cells. But T cells in the tumor, they can come in different flavors. There are different subtypes and states of T cells in the tumor. And very often, T cells in the tumor, they display an exhausted phenotype, which is very similar to T cells found in during chronic virus infection. Those cells they express high levels of PD-1, TIM3, and LAG3, and they are also reported to have impaired effector function and also impaired proliferation capacity. But what we found out with our technology is that those cells are actually actively proliferating, which I will talk to you later. So another question about T cells in human cancer is, what is the relationship between different subtypes or states of T cells? Could one population differentiate into another? So that's actually the motivation of why we developed this technology. So we use this technology to profile immune cells from 25 melanoma patients, and then we profile all the immune cells at transcriptome level, single cell transcriptome level, and also for T cells, we obtain their T cell receptor sequence and also transcriptome sequence. So how does the T cells in human melanoma looks like? So those are the cells we sequence, all the T cells we sequence. We sequence around 30,000 T cells, and then we use an algorithm that we developed called MetaCell to cluster those single cell transcriptome profile into those 218 metacells. So basically, this pipeline, they can cluster the transcriptome, single cell transcriptome based on their similarity. And one metacell is basically a group of highly similar single cell transcriptome profile. And then based on the similarities of those around 200 metacells, we can further merge those metacells into a bigger group. So we are able to identify NK cells and naive-like CD8 T cells and regulatory T cells expressing very high levels of FOXP3, also follicular helper T cells, and also memory CD8 T cells and dysfunctional CD4 T cells, and also cytotoxic CD8 T cells, and dysfunctional CD8 T cells, or people often call them exhausted CD8 T cells. And we also identify an interesting population which is lying between the naive-like population and also the dysfunctional CD8 population. The dysfunctional CD8 population, they express very high levels of PD-1, LAG3, and also TIM3, TIGIT. And interestingly, they also express very high levels of interferon gamma, at least at the mRNA level. So from the 2D projection, we can appreciate that the T cells, particularly for the CD8 T cells, they are actually quite from the transcriptome behavior, you can observe there's kind of a transcriptome gradients among different CD8 T cell subtypes. They are kind of intermingled with each other. For example, naive-like and the transitional and the dysfunctional CD8. So such behavior indicates there's a active differentiation process going on in the tumor for CD8 T cells. So to further investigate the potential differentiation process of CD8 T cells in the tumor, we then take all the CD8 T cells and look at the gene behavior of the CD8 T cells. We can calculate the gene correlation of the CD8 within the CD8 T cells and identify co-expressed gene modules in the CD8 T cells. We managed to identify a few interesting modules. For example, a cytotoxic module, which is featuring granzyme A, K, and GANI, and granzyme H. We also identify a very familiar gene module. We call it a dysfunctional gene module, featuring LAG3, TIGIT, and PD1 and TIM3. So basically, the cytotoxic module, it defines what we mentioned earlier, the cytotoxic T cell class, and that dysfunctional module defined a dysfunctional CD8 T cells. And interestingly, we found that the two gene modules are actually anti-correlated with each other, meaning the two gene programs they are antagonizing each other. This indicates that CD8 T cells can either differentiate into cytotoxic T cells or into dysfunctional CD8 T cells. So to better quantify this process, we take out all the genes from the dysfunctional gene module and also all the genes from the cytotoxic gene module and quantify the expression level of those genes from the two gene module in all the CD8 T cells. So the right panel is a plot showing the dysfunctional score, meaning the expression level of the genes within the dysfunctional gene module, and also the y-axis is the cytotoxic score for all the CD8 T cells. Each dot here is a meta cell. It's not a single cell, but a group of highly similar cells. And the color here indicates their group. We have the I'm showing here transitional population and also dysfunctional population and cytotoxic CD8 population. From the dysfunctional score and cytotoxic score plot, you can I think it's clear that the transitional and the dysfunctional, they are kind of connected, while the cytotoxic population score are not connected to the transitional or dysfunctional population. So this behavior indicates that the transitional population can indeed differentiate into dysfunctional population. And if that is true, then the two populations should share the same T cell receptor sequence. So we then calculate the T cell receptor sharing between different T cell population. So the value I'm showing here is if the value is positive, that means the two population is share T cell receptor. If the value is negative, that means the two populations they do not share a T cell receptor. So we found that indeed a dysfunctional population share T cell receptor with a transitional population, but the cytotoxic population they do not share a T cell receptor sequence either with the transitional population or with the dysfunctional population. So I showed you the cellular heterogeneity of T cells, and also our data support such kind of differentiation process of different T cell subpopulation. Based on the transcriptional gradient and also TCR sharing, we think in human melanoma there's a transitional towards a dysfunctional state differentiation process. However, the cytotoxic population that we found in human melanoma, they are not connected to this differentiation process. We all know that the immune repertoire is, as Hanjie mentioned earlier, the immune repertoire is it can indicate the disease state, and also it can indicate the response rate to immunotherapy. So we then look at the T cell clonal expansion state for those patients. And we found that the T cell clonal expansion can be quite variable between different patients. For example, in some patients, in patient 11, we don't really see a T cell clonal expansion. Those are the T cells from the tumor, not from the peripheral blood. So this indicates there's very limited a T cell response within the tumor. However, in patient 28, you can observe a very dominant clone. There is around 40% of the T cells, they can share the same T cell receptor sequence, indicating that there's a active T cell proliferation, a clonal expansion in this patient. So we then ask what is the difference between the T cells that underwent clonal expansion versus those that are they did not undergo clonal expansion. So for each patient, we then separate the T cells into two groups. One group is T cells that underwent clonal expansion. The other group is T cells that did not undergo clonal expansion. And then we compare the T cell subtype composition between the two groups. And I think it's very clear from the plot that you can see that the purple population, the dysfunctional population, the percentage of the dysfunctional population is much higher in the clonal expansion group, and this is true for all the patients. So this indicates that the dysfunctional CD8 T cell are actually they form large clones in human melanoma. To further understand this process, we ask if dysfunctional CD8 T cells are the cells that form large clones, then we should see much higher T cell clonality in patients that have higher levels of dysfunctional CD8 T cells. So then we order all the patients based on the percentage of dysfunctional CD8 T cells, and then correlate that with T cell clonality. We see a very good correlation between the percentage of dysfunctional cells and T cell clonal expansion. So those data actually support that the dysfunctional CD8 T cells, indeed, they are undergoing clonal expansion. At least, I mean, form large clones in the tumor, but that doesn't mean they are proliferating within the tumor. So the other possibility is that they could proliferate before they migrate into the tumor, and that's why we found this many different dysfunctional T cells sharing the same TCR. So to understand which scenario is correct, we then further analyze the transcriptome signatures of those cells. So one of the advantages of single cell RNA-seq is that it is able to detect whether a cell is undergoing proliferation or not, based on the expression level of genes that are related to a cell proliferation. And we then calculate the percentage of proliferating cells within different immune subtypes. We found that the dysfunctional CD8 T cells population, they have the highest level of proliferating cells. We also validate that with conventional flow cytometry analysis. So basically, what we think is happening is that the dysfunctional CD8 T cells, they are actually actively proliferating within the tumor, and that's why they form large clones. So I showed you the cellular heterogeneity and how T cells differentiate in the tumor, and also the interesting finding about active proliferation and clonal expansion of of dysfunctional CD8 T cells. But what about their function? Which T cell subpopulation is tumor specific? Which population is bystander, is not tumor specific? So to answer that question, we along with the single cell profiling, we also expand the cells that we obtain from the patient, and then co-culture the expanded T cells with a tumor digest from the same patient. And if the expanded T cells, they contain a tumor reactive clones, then those T cells will be activated and start to secrete interferon gamma and TNF alpha. So using this assay, we can we analyze samples from 10 patients, and you can see that in some patients, for example, the last 4 patients, we could not detect any tumor reactive T cells, but in other patients, for example, in patient 9, we can detect up to 40% of the T cells can be activated by the tumor cells from the same patient. So we then compare the tumor reactivity data with the T cell composition of T cell signatures of different patients. First, we order the patients based on the percentage of dysfunctional CD8 T cells, and you can see there's an anti-correlation between the dysfunctional the percentage versus the cytotoxic percentage, which is consistent with the gene module analysis. And when we correlate this signature with the tumor reactivity data, we found that all those patients that have tumor reactive T cells have high levels of dysfunctional T cells, but very low levels of cytotoxic T cells. But for those that do not have tumor reactive T cells, they have very low levels of dysfunctional T cells, but high levels of cytotoxic T cells. So this data indicates that the dysfunctional T cells are the tumor specific ones. However, the cytotoxic T cells are bystander T cells. So a few papers actually trying to identify the markers for tumor reactive T cells. For example, they found CD39 and CD103, and the AIRR-1 is actually a marker for bystander CD8 T cells. So we then analyzed the expression level of those 3 markers in our CD8 T cells. We found that the tumor reactive markers CD39 and CD103 are highly expressed in the dysfunctional CD8 population, are lowly expressed in the cytotoxic population. The bystander marker AIRR-1 is highly expressed in the cytotoxic population, but lowly expressed in the dysfunctional CD8 population. So our data actually is actually indicating that the ratio of dysfunctional CD8 T cells versus the cytotoxic T cells could potentially be used as an indicator of whether there's tumor reactive T cells in human cancer. So I would like to summarize my talk. So we have developed a new technology to profile the immune infiltrates, particularly for T cells. We can obtain their transcriptome and also TCR sequence that allow us to really follow the T cells and to understand their clonal behavior and how they differentiate in a disease state. And using this technology, we identify actively proliferation process of CD8 T cells from the transitional state towards the dysfunctional state. And we identify the transcriptome signatures of dysfunctional CD8 T cells, and they are actively proliferating, and the abundance of dysfunctional CD8 T cells is associated with tumor recognition. And with that, I would like to thank my postdoc supervisor, Ito Ami, and also my collaborators. And I just started my new lab 5 months ago. So we are open to, we have many positions at different levels open for, if you are interested in my lab, so please feel free to contact me, and we can chat further. Thank you very much.
Very perfect timing. I'm wondering whether there's any questions. Okay, we don't have too much. I think just a very short question. I'm wondering that besides alpha beta T cells in the melanoma, have you ever tried to investigate the gamma delta T cells?
No, we didn't, but I think we didn't detect much gamma delta T cells from the tumor biopsy.
Okay, thank you.